Okay, good afternoon everyone. My name is Alex Golberg and I'm a research fellow at the Shriners Hospital for Children in Boston, in Massachusetts General Hospital and in Harvard Medical School. And today we're going to talk about the use of pulse electric fields for burn wound disinfection and the use of QuickField software for modeling of the electric field distribution inside the skin. So the motivation behind this project is in burns, and fire and burn injuries represent about 1% of total injuries globally. In the United States in 2010, there were about 20,000 injuries of the fire, which eventually lead to the contamination of the wound. And wound sepsis is the major cause of death in the burn injured patients. So in their recent 2013 study concluded that there is no clear evidence of efficacy of any antibiotic treatments of the wound. So eventually all the drugs for the wound disinfections we have now are not very effective, and currently there is no effective therapy for burn wound disinfection. So the hypothesis behind our project is the non-thermal pulse electric fields kill the bacteria in the wound by irreversible electroporation of the cell membrane. And here is how the process theoretically looks like. We have an intact bacterial cell, and once we apply the electric fields, there is a formation of the pores in the bacterial cell. And if these pores are irreversible and they are big, bacteria die. So the phenomenon of the permeabilization increase of the cell membrane wall by pulse electric fields is called electroporation. Now, what is electroporation? Electroporation is, as I mentioned, is a phenomenon where the pulse electric fields increase the penetration or the permeabilization of the cell membrane to external substances. Now, on the left, we can see how this looks like. So this is the cell membrane, and the yellow and bluish molecules are phospholipids, and here we use the multimolecular simulations for this. And what is exhibited in the pink color here are the water molecules. So once we apply pulse electric fields, the water molecules from both sides of the membrane create water bridges. So we can see them on exhibit B. And these bridges actually form the pores or aqueous pores. So it's thought that electroporation leads to the cell death by the creation of multiple of these pores on the cell membrane. Now there are many applications of the pulse electric fields in medicine and in industry. Recently years there are two young therapies which emerged for the cancer treatment. One of them is the irreversible electroporation or nanoknife, which is used for solid tumor ablation. And in that case we just position the electrodes on the tumor, apply pulse electric fields, and kill the tumor that way. The second treatment is called electrochemotherapy. In electrochemotherapy, pulse electric fields are used to increase the permeabilization of the tumor cells to the drugs, for example, blitomycin. So we position the electrodes on the tumor, we inject the blitomycin, apply the pulse electric fields, and then the blitomycin goes inside the tumors and kills the cells. The third application in medicine is the DNA vaccination, and you can see it's here. This is the gun, which is used for the penetration of the DNA molecules inside the cells, mostly on the skin, and now there are a couple of clinical trials which use that technology for multiple diseases. And finally, electroporation of pulse electric fields are also used in industry, and on the right bottom exhibit you can see the large 100-ton-an-hour plant which uses pulse electric field to increase the permeabilization of the sugar beets for the sugar extraction processes. And these processes save about 20 to 50% on the energy. So given all these applications, we decided to try to use the pulse electric fields further in medicine, but for the disinfection purposes. However, this idea is not new. The pulse electric fields for the disinfection emerged from the food industry starting from the 1961 when the first patent emerged. And now, as we can see on the picture here, there are large processes available which kill the bacteria inside the food by the application of pulse electric fields. Now, the FDA recently issued the guidelines to the industry on how to kill specific bacteria in the foods. So given these applications of pulse electric field in medicine for solid tumor ablation and DNA vaccination, and the ability of pulse electric fields to disinfect food, we decided to try and kill the bacteria on the skin and inside the burn injury. So our preliminary study we did on the eradication of multidrug resistant Acinetobacter baumannii in the burn wounds using pulse electric fields. And what we did, we used first the numerical model in the QuickField for electric field distribution modeling inside the burn injury in the mice skin. And then we also did the experimental model on the black C57 mice with infected with the Acinetobacter baumannii. So here we can see schematically our model, which we tried in the QuickField. So the geometry appears in Exhibit A, and we can see here the pinch of the skin. So this is the skin, and we have two electrodes around it. Now, skin is a complex tissue, this is important to remember, and it has multiple layers. And we modeled all these layers, starting from stratum corneum, here externally, then there is an epidermis, dermis, and panniculus carnosus, which is a skeletal muscle inside the skin in mice. However, once we induce the burn to the tissue, all these layers are gone. Eventually, each layer has its own electrical conductivity, but once there is a burn, all the membranes, everything inside the tissue are destroyed. And eventually, we can look at the burn as a homogeneous tissue, which appears here is a burned and infected tissue. Now, our treatment we deliver through the electrodes, and so here we can see the model of the electrodes. Now, we first modeled the distribution of pulse electric fields inside the skin. And why this is important? It is because all other currently available techniques, be it drugs or lasers, have a very limited penetration inside the skin. So drugs are limited by the diffusion and lasers are limited by the scattering, and they never get the full thickness distribution of the same dose of the treating reagent. And QuickField actually enabled us to show that once we apply the pulse electric fields on the surface of the skin, we get a pretty homogeneous distribution of the field. And we can see here two models. One model, the first one here on the left, Exhibit B, shows what happens to the field once we apply 500 volts on one electrode and 0 volts on another one. And we can see that the distribution of the electric field was pretty homogeneous, including the ages. Once we apply with 1000 volts, we see that fields are higher, but still the distribution is homogeneous. So the cells inside the epidermis, dermis, and in this case the burned and infected tissue are exposed to the same electric fields. And this is important in the cases of deep infections once the bacteria leave not only the surface of the skin, but once they go deeper, and we want to kill them there. And again, it's not possible doing with lasers or drugs today because of the limited penetration. However, the question which we also get always is if this treatment is really non-thermal. And so using QuickField again, we modeled the coupling problem of the electrothermal effects of the pulse fields. So here are just the results. We have our skin model, and this is our skin flap again in the center, and then we have the left and right electrodes. And once we apply 1000 pulses, we can first see the electric field distribution, and next we can see the temperature distribution. So it's important to remember that in our case, we have a body on the which is there, which actually heats the tissue from the bottom, and we have the air from the top, which cools the whole system. Now, extracting the temperature profiling after the application of 80 pulses, we can see the following results, which are very interesting. So first, we can see the temperature distribution as a function of the location in the skin flap. And again, all this was extracted from the QuickField. So this is the black line on the Exhibit A, and after 80 pulses, we can see how the temperature is distributed there. So we have a almost body temperature at the bottom, but then we have the increase on the temperature inside the tissue with a maximum in the center. So this maximum, we modeled it separately with a black dot here, and we found that the temperature in the center increases very fast after about 20 seconds or 20 pulses as the treatment was delivered in 1 Hz, and then we just have it stabilized. Now the stabilization is because again of the air cooling and of the body framing from the other side. So QuickField eventually enabled us to model this process of the pulse electric field distribution inside the skin, and also show that the phenomenon is non-thermal, because 41 degrees of final temperature, as we can see it here, just cannot kill the bacteria cells. So next we did the experiment, and here how the experiment looked like. So we have the burn model on the mice. So this is the mice. We apply the burn model by 95 degrees heated electrode for 10 seconds. So here is how the burnt area looks like. And then we infected it with Acinetobacter baumannii, the multidrug resistant bacteria. So in the orange color here we can see the frame of the distributed where the infection took place. We treated the infection using pulse electric fields as appears here, and this exactly resembles our model as we showed that before. So we have two electrodes and the skin flap in between. Finally, we used the Acinetobacter baumannii, which is in our case was a bioluminescent strains of bacteria. So we could quantify the number of bacteria in the wound using the bioluminescent imaging, as we can see it here. So the lighter, the more strong the illumination area is from the specific area, more bacteria are there. And eventually what we hope is that application of pulse electric fields will destroy the bacteria. Now the whole experiment design appears on exhibit C here. So at time 0 when induced the burn in the mice. 5 minutes after, we left for 5 minutes to cool. We infected the burn with Acinetobacter baumannii, bioluminescent. So to show that there is a real infection, we made the imaging in 10 minutes after the burn, and then we left it for 30 minutes. And we left it for 30 minutes to see that infection takes place and the bacteria is not swamped away and doesn't disappear. So at 35 minutes we performed another imaging, so to be sure that the infection is there, and then we treated the wounds for the first time with the 40 pulses. Then we performed an imaging and we treated them again, another 40 pulses, so total each mice was treated with 80 pulses. And we performed the imaging and then we waited for 3 hours. And we waited for 3 hours to be sure that the infection doesn't come back, or to test if it does come back. So in our case we tried 2 different doses of treatments. We applied 500 volts and 1000 volts, and in both cases we used 70 microsecond pulse length and 1 Hertz, and it was mentioned we first applied 40 pulses and then we applied 80 pulses. So in the low dose of treatment, which we can see on exhibit A here, we see that immediately after the infection we can see this strong signal on the mice back, and 35 minutes after the signal wasn't lower, so still the infection is there. Application of 40 pulses caused to the significant reduction of the signal from the mice, and we got about log 2 reduction. And application of 80 pulses caused another reduction in the bacterial load, which led to almost log 4 reduction in the bacteria count on the back. 1000 pulses caused a more significant reduction immediately after the treatment, which can be appreciated here from the 45-minute and 55-minute pictures, images. So the 40 pulses reduced in luminescent bacterial load for log 2, and the 80 pulses reduced them almost for log 5. So we can see the immediate effects of the pulse electric fields on the Acinetobacter baumannii are drastic, and we reduce a lot of the bacteria immediately. And then the question is if they come back, so that's why we waited for 30 minutes. And we can see the results here. So in the control section we just wanted to see what happens with the bacteria itself without any treatments 3 hours after the infection. So some of the bacteria eventually die, or they just go into a deep infection inside the tissue. So we can see the reduction in the signal from the bacteria, as we can see here in the control. So it's about log 1 reduction. Application of 500 volts, we can see that immediately after the treatment we got log 5 reduction in bacteria, but then 3 hours after some of them grew back, as we can see them here. And eventually 3 hours after we achieved about log 3 reduction. And 1000 volts application of 80 pulses caused to log 5 reduction 3 hours immediately after the treatment, and the same log 4, log 5 reduction was observed 3 hours after the treatment. So our conclusions are that using 80 pulses with a 500 volt per millimeter, in our case it was 1000 volts, we achieved a stable disinfection with a almost log 5 reduction of the multidrug resistant Acinetobacter baumannii, and these results were tested 3 hours after the treatment. So importantly, our numerical model suggested that the pulse electric field penetration is homogeneous throughout the entire segment of the infected tissue, and the pulse electric field's effects on the bacterial reduction are non-thermal. So I wanted to acknowledge the Shriners Foundation and MGH for the support of this study, and the study was performed in a large group of scientists as appears here. And now I want to go to the QuickField and to show you the procedures we did, and also the software I hope will be available on the QuickField webline. So in the QuickField, in order to model these three phenomena, we need to use we actually need to create three models. The first model, which appears here as DC1000, is the model where we solve the Laplace equation for the distribution of the electric field inside the tissue. And the problem properties are: this is a DC conduction model, plain plate with the depth of 2000 microns. So we can see here that the precision is normal, coordinate system is Cartesian, and the file name is DC1000. So we use the same geometry throughout all our three models, and in this case the geometry file is called -geometry-adjusted.mode. So eventually, once we want to create a new model, this is how it looks like. So this is a simple DC conduction model. Next, we drew the geometry, and here we can see the geometry of our skin flap with the electrodes. So in the center here, all these is the geometry of the skin. And as I mentioned, skin is a complex organ, so we have multiple layers. Now let's go just a bit closer to see how it looks like and what are the abilities of the QuickFields. So as you can see here, we have all layers. On the left, we start with the electrode. Next, we have the stratum corneum. We have epidermis. We have dermis. Then we have muscle, and we have subcutaneous tissue. So we got this resolution of the skin model, and this is how we draw it. So the next step will be to give the labels. Now, in our case, we give labels both to blocks and to edges. And let's start with blocks. And I mentioned we have here dermis and infected dermis. So all area which is outside the gonad infection, all this area, is the normal skin. Normal skin has its own properties. Inside the treated area and the gonad infected tissue, all these properties are destroyed because of the burn, and they all have the same ones. However, to get the distribution of the electric field inside the tissue, we need to give this as initial conditions. So here is the dermis. As I mentioned, dermis is outside the burn area, and this is normal dermis. And once we push down here, we get the electrical conductivity properties, so we just put the numbers from the literature, and in this case it's an isotropic. It's isotropic tissue with the 0.2 Siemens per meter electrical conductivity. Now the burned and infection dermis is different. Now this data was measured by us in this experiment, and it's about 0.13 Siemens per meter electrical conductivity. Electrode is it's conductive medium, but it's just outside. We don't have any specific properties. The epidermis has 0.2 Siemens per meter. Epidermis in the area of the burn has 0.13. The muscle, normal muscle, has the electrical property of 0.4 Siemens per meter. Electroporated and infected muscle has 0.13, again measured in our experiment. Stratum corneum, normal one, has a very low electrical conductivity. So this we just cannot see it because the layers are very small, but if we zoom in, we can just go up, go out a little bit, so we can see them here that this is the stratum corneum, very thin layer, and its electrical conductivity is very low. Stratum corneum in burned and infected tissue is destroyed eventually, so we have again the electrical conductivity which is higher in this case, 0.13 Siemens per meter. And then we get the subcutaneous tissue, which is here, and again we have the normal subcutaneous tissue in the affected area, and we have the damaged subcutaneous tissue in the affected area. And the properties again are different. Again, the subcutaneous tissue in the affected area has a 0.13 siemens per meter. Okay, so now we gave the basic properties for the materials in the model, and there's a composed model because we have a lot of different materials here, both normal tissue and injured tissue. And the second part of the process will be to give the boundary conditions, and the boundary conditions we give on the in the age labels. So it's here. Left electrode, we put the 1000 volts boundary condition, and here's our left electrode here, just enhanced with red. Our right electrode is grounded, 0 volts. Actually, that's it. That's that easy. So we just need to draw the geometry, and this part usually takes time, specifically in this case. Then we need to give the the properties of the material, the electrical conductivity in this case. Next, we need to give the boundary conditions. So in this case, again, it's how much voltage we apply for each electrode. Then we need to mesh. So in this case, we already meshed it, but in order to mesh, we just push this button. So it's called build mesh, then we get mesh everywhere. And finally, we just push the solve button, which is here. So now today, do you want to solve a problem again? Yes. And now we just need to wait a bit until we get the solution. So we're still waiting for the solution. And yes, we want to see the results. Okay, here are the results. They show the current density, but what we want to see the field. That's why we go to the view field picture properties. Here we want to see the field strength. Now we're interested in the specific field, so let's go to the describe minimum and maximum field we want to see. Oh, that's nice. So eventually we see the whole profile of field distribution inside. Then again, if we want to increase the resolution, we go again for the field properties. This one can increase to 3. So this is the process of optimization, how exactly we get the numbers of the field and the colors as we want. So we can play with that and show the different configurations of the field distribution. So the strength of the field appears here. Here is the legend, and here just the color, we can see how much is there. So again, if we want to improve it a little bit, we can just reduce it. Yeah, here is nice. So we can see exactly how what the field is in which point. Now, an interesting property here is the local display. So this is this button. So puncturing on every point will give us the electric field in the specific location. So we just want to probe the electric field inside the epidermis, inside the dermis. So it gives us the field strength just up here. If we want to probe it another point inside the muscle, it's here, or we probe it inside the subcutaneous tissue, it's here. So eventually the numbers here change as we go to different areas. But if inside the electric field, eventually. So this is a nice tool which gives us an opportunity to probe the electric fields inside the tissue. Now we want to see also the distribution of the field. And how we do that, we use the function. Go on the properties. So to do it, we just need to draw the contour through which we want to see the field, and the contour is here. So just push it here, and we just go in. And for this, we just use this XY plot. So as now the electric field strength as a function of the location. So we can see that there is nothing, and then suddenly we have this large electric field which is very homogeneous, and then it goes down again about the end of the tissue and goes to zero. So here we can check what we see. We want to see electric field strength and in which direction. Again, we can also see the conductivity, and we also can see the temperature. But temperature is not this part of this problem. Next, we wanted, so actually what we saw now is both parts which we showed in the presentation. So how we can calculate the temperature in the spot using this function of the local value. So we just go and punch it here. Or how we can calculate the electric field distribution along the tissue using the contour function, which is here. Another interesting property is the table. So I actually use it a lot because pushing the table gives us all the properties inside along the contour, which include the geometric location, the voltage, the electric field, the currents, other things we're interested in. So we can just export it from here directly to Excel or MATLAB and then to process the data separately. So to conclude, in this part of the seminar, what we did, we draw the model of the mice skin, punched between the two electrodes, applied the voltage on one of the electrodes, and investigated the distribution of the electric field inside the area using localized function or using the control line. So this actually covers us the first part of the problem of the homogeneous distribution of the electric field inside the tissue. So again, we can appreciate here again that the distribution is pretty homogeneous. The next part was to solve the electrical thermal problem and to see what's the heating effects of the pulse electric fields on the tissue. After we solve the electric field distribution problem, we want to see now how the heat transfer equation, what's the effects of the pulse electric fields on the heating. Now, to the heating, we need to solve two equations. One is the stationary heat, where we give the initial conditions and where we heat up all the tissue to our initial conditions. And the second one is the transient heat to see what happens to the tissue once we apply the pulse electric fields. So for the transient heat, we solve the steady-state heat transfer problem. Again, we use the same geometry, my skin geometry adjusted mode, and it's very convenient, so we don't need to redraw it. We just can use the same geometry we used for the electric field before, and we use the steady-state heat transfer problem. Now, the idea here, so in order to calculate the initial conditions, we need to know the power. Again, the blocks, and here we give the electrical property of our tissue, thermal conductivity in this case. So again, all this data is from literature, and we need to go one all the blocks and just get the thermal conductivity input of the dermis, and in this case the thermal conductivity of the burnt and injured dermis. Again, an electrode, it's a metal, so it's like very large conductivity, and so forth. Now, importantly, we need also to get the boundary conditions because we have an open problem. In our case, we have air, and the air will give him a temperature. We have a body on the bottom here with the red lines, so we'll give it a temperature, like it's 310 Kelvin in this case. Right and left electrodes, again, that's the temperature of the air. So, and once we solve the problem, we'll actually give the initial conditions for our system. And again, these are the conditions before they apply the electric fields. Next, we need to go and solve the transient heat problem. Now, in transient heat problem, it's a bit tricky. So again, we go to the model properties. And now what we need first to, again, define this is a transient heat transfer problem. Geometry is the same geometry we used previously for the DC conduction, where we showed the electrical field distribution. But here we need to import the data. We need to link it to our initial heat conditions. And this is here. So as you can see here in the data sources, we need to link our transient heat problem to the DC conduction problem, which shows us how much heat is generated, and to the temperature field, to the initial temperature conditions. And here, this is done here. So, and again, finally, the timing, we need to set for how much time are we heating. So again, to define this problem, we need geometry, we need the electrical input, how much power we are applying in, and then we also need the initial conditions of the temperature inside the tissues. And in this case, we just used this linkage process. So we can see that we can connect our transient heat problem to the DC conduction and to the initial heat problems solved previously. And so once we get this, again we go and define our properties, electrical, our thermal conductivities, all these are imported from the initial heat condition. Dermis, epidermis, electrode, muscle, and so forth. Just check out that we really get all of them. Start on corneum, so we just need to enter them one by one, all the thermal properties. Okay, we can get them. And again, the labeling, so we have this air conduction. Now the heat transfer with the air is by convection, and which we can define here. So in this case, it's important that the air temperature is not constant. It was in the initial conditions, but we have this convection properties, and this is again from the literature. And the body, we have a constant temperature because it's conduction. And that's it. So our links, we can see them here. So these are the links to the initial problems, which give us the power, how much electricity we transfer through the tissue, and also the initial properties of the initial heat properties of the tissue. How we solve the problem. Okay, great. And so here we can see the results. Results give us the temperature distribution inside our piece of skin and the electrodes. And again, the color map is here, gives us the temperature, legend on the right. Now we can see the temperature increase over time. So if we go here, we see that this is at the 80 seconds, so this is actually how it looks like after the last pulse. And here we can see the resolution. So this is the initial, like 8 seconds after the initiation of the electric fields. So we see that these things are heating up. Here is in the middle, for example, 40 seconds after the application of the electric field. It's really nice that we can see the dynamic process. So this is how the temperature distribution looks like 40 seconds after. And if we go back to the 80 seconds, so that will be the end of the process. So we eventually can investigate the whole dynamics of the tissue heating by the application of the electric fields. And again, we can probe different temperature at different locations. So as mentioned before, the largest heating is in the center, so we just push the button here, we get the temperature about 312 kelvins. And if we go outside the electroplated area, we see that it's about a body temperature of 310 kelvins. Additionally, we can again investigate the temperature using our contour property. So we just draw a line here, and then we just calculate it. And here we get the temperature distribution alongside the contour. So this is our skin, the temperature distribution inside our skin flap. So the electrodes, they're not heated, and then we have the maximum temperature in the center, so there is an increase, and then again it's a decrease in the temperature, so we can just see them together. It's very convenient property. Now, in addition, if we want to export it to Excel, we can go to the table view. So the table view again gives us the location, L here, and then we get the temperature in a specific location, and in addition to other properties and fluxes which change over that. So again, we just demonstrated the combined electrothermal problem and its solution in the QuickField. For this, so it's modular and very convenient. So we solve separately the initial conditions of the temperature problem, then we can on that add different transient conditions, that we can see the solutions at each point, and also distribution in the space and time. So this is the space distribution of the temperatures, and again, if I come back, show here, this is the time distribution. So this is how it behaves over time. So I said it was very nice to show in the real time how we can just, in 45 minutes to draw the problem, which is a complex electrothermal problem, and get the solutions which are actually very relevant to the treatment planning, because now once we have these models, we can apply different parameters and see how they affect our treatment plannings of pulse electric fields for the disinfection. Oops. Okay, now I just exit QuickField and go back here just to show you again that we did the modeling of the electric field distribution inside our skin flap using all the properties of the skin at different voltages applied. We did the thermal effects on the skin, so we actually monitor the temperature and how the pulse electric fields affect the temperature, and we see that the heating is up to 41 degrees. And then finally we did the experiment where we did exactly the same geometry. We applied the 80 pulses during 80 seconds, and we killed the bacteria, reducing more than log 5 of the bacterial load of multidrug resistant Acinetobacter baumannii. So again, this is the immediate effects of the field, and this is the long-term effects where the log 5 reduction was achieved. QuickField enabled us actually to see to model the process and to show that the treatment is homogeneous and that the pulse electric fields effects on the bacteria are non-thermal. And with this, I want to conclude the seminar, and thank you very much for listening. And if you have any questions, you can always ask me. So this is Alex Golberg from Mass General Hospital. Thank you.